Good morning and good afternoon to all of you attending. We would like to welcome you to day one of our Forensic Toxicology webinar week, where the first presentation will be going over a practical high-resolution mass spectrometry screening approach and propositions for identification criteria, quality assurance, and validation. This will be presented by Dr. Sarah Wiley. Please use this webinar console in order to ask any questions that you may have. And then also included on this page, you'll see that there are resources that you can click on to get more information. And then there's also a section off to the right that you can see that has the speaker bio information. And there's a link to get more information on our presenter today. And then underneath that, you'll see a webinar survey. If you could please take the time to fill that out, we would greatly appreciate it. Thank you. Our first presenter today is Dr. Sarah Wiley. She is a judicial expert in toxicology from the National Institute of Criminalistics and Criminology. She is registered as an expert in forensic toxicology. She obtained her PhD in analytical toxicology at Ghent University, and her current interests include novel psychoactive substances, alternative matrices, drugs and driving, and method validation. Sarah is a board member of TAFT and also BLT. She has also obtained the TAFT Achievement Award. We are very honored to have her here as our first presenter. And with that, I'll hand it over to you, Dr. Wiley.
Thanks for this introduction. So good evening, good afternoon, good morning, wherever you may be looking into this webinar. I'm Sarah, and I will be talking about a practical screening approach today using high-resolution mass spectrometry. Now, as a forensic laboratory dealing with analysis of various biological matrices for the judicial authorities, we of course want a very practical screening approach, and today I will be looking a little bit more into the identification criteria that we use, but also quality assurance aspects and some validation specifics. According to the European Drug Report, drugs are used everywhere by everyone and can be everything. And this is what we have to deal with, in the sense that we have to monitor classical drugs such as, for example, cocaine, MDMA, amphetamines. But when we look into this report more in detail, we of course have lots of new psychoactive substances to monitor. And each year, new ones are still popping up. For example, according to the report of 2022, we have to monitor 880 NPSs and 52 ones are new ones from 2021. So this is an enormous number of compounds that we have to be able to detect, so we need broad screening approaches to tackle this. Just to give you a view of what we see in Belgium, I put this slide together. So we have lots of what we call classical drugs such as cocaine coming into our ports, but we also have various production sites of MDMA. And this is a Belgian map where you can see that our drug unit has found illegal laboratories producing MDMA, but also dumping sites. Now, besides, I would say, the classical MDMA pills, we can also find pills such as this one. So the green woman was a pill containing 2-CB. We also find powders in which we see mixtures of ketamine together with MDMA and 2-CB. And then more and more in our routine casework, we observe designer benzodiazepines. Concerning the synthetic cannabinoids, we have to know that in Europe alone, in 2021, there were 15 new ones, and in total, there are 224 compounds. We don't observe them that much in our casework, but we do observe snippets of paper in a more prison setting in which synthetic cannabinoids are sprayed onto the paper and then distributed via snippets to be smoked. Just to summarize with a famous movie quote from Forrest Gump, I would say that sample analysis is like a box of chocolates. You never know what you're gonna get. So the challenges are really the wide drug market. We have a whole range of compounds, and this includes a high turnover, because what's popular today might not be popular tomorrow. And this, of course, has an impact on how you will develop your method, which compounds do we need to detect, and certainly when you think of other demands, such as, for example, costs. Because if you want to buy all of the reference material, this can have a budgetary implication. Other clients' demands that we have to take into consideration when developing a method are, of course, a kind of time frame of report. When we have to do an analysis for the judicial authorities, time is limited, so we need a kind of fast and adequate screening method. As we have an accreditation via the ISO 17025, it's also important to have a good idea about the risks that we have with our methods, in the sense that we need to demonstrate, of course, that there's an adequate selectivity and so on. So we need to have a good idea about how are we going to tackle validation and quality assurance. And this is really our aim. I want to talk about a kind of high throughput, high resolution mass spectrometric screening, how we looked into validation and the quality assurance. So the method that we apply is, works on a G2 XS time of flight from Waters, and we use the Unified Forensic Toxicology Screening application using an Equity UPLC HSS C18 column. Now what actually happens is that when we get samples in, we like to have an initial screening on our time of flight. We use 100 microliter of sample, and this can be blood, urine, but also vitreous humor, oral fluid, or for example stomach contents, which of course will first be diluted. We will do a sample preparation consisting of a protein precipitation with 400 microliters of acetonitrile, and we wanted to keep it as simple as possible because we just want to monitor or screen as much compounds as possible. When we applied the original Unify method, we observed that for some synthetic cannabinoids, they eluted rather late, and so they were actually kind of stuck in the washing phase at the end of the chromatogram. So we did a longer gradient from 15 to 21 minutes to, observe the synthetic cannabinoids more easily. We not only used Waters library, but also the high-res NPS one, which consists of 2,160 compounds. Up to date, we don't have retention times for those in this library, but we're planning on doing so. Now, very important thing is looking into identification criteria. Certainly when you use a large database, you can have the possibility of having false positive hits. So identification criteria are really of importance, and this was already established in some guidelines, for example, the European Commission decision of 2002-675, or for example, the Australian and New Zealand documents, but it was also looked into by the American Academy of Forensic Science Standards. So you have to have a kind of identification criteria which is based on, of course, accurate mass, but also on retention time, or for example, fragment ions and their relative abundance. And so what we did was we looked into identification criteria such as mass error should be lower than 5 ppm. We look into abundance of fragments via isotope match, but also intensity, retention time, and then we look into response. We actually divide it into two subcategories. If it's larger than 10,000, this will give us, a good idea of interesting compounds in our routine work. The fragment ions, of course, that we expect and are found are also part of our identification criteria. So here you see an example. In this blood sample, we found an N-bone. Now, you can also see on the left-hand side that our system suggested more compounds. So that's why we really need good identification criteria to have a quick selection first of possible compounds. In the way that we work, we will always do an extra confirmation via an LC-MS method with reference materials. So in this case, we would select the N-bomb, which is then confirmed via a standard. But for example, when you look at the second line, we can see that the isotope match intensity is really not according to the criteria, and this is considered as a false positive. From our drug unit, we get drug seizures. Now, in this example on the slide, we had 49 different substances, most of them stimulants. Now what happens is that we will inject them into our system, and sometimes we don't know the identity. So what happens then is that our drug unit has different methods to establish the identity, and we will add these compounds to our system, improving the final method for future screening purposes. This is the same slide, but actually different example. Here we're talking about designer benzodiazepines, and again we will inject them, look into the fact if the chromatography works, if we can either detect them, or if we have to include them in our in-house library. And then, of course, validation of the methods. Now, maybe a bizarre start of this section of the presentation, but I think it gives you a very good visual of what we mean with validation. It's really necessary. We can have all kinds of expectations or ideas of our methods, but we should really know the reality. What is our method really up to? What can it monitor? In which, sensitivity? Is it selective, or do we think it is? What can we actually do with it, and what are, the pros and the cons of this method? So validation is really all about looking if your method is fit for purpose. And I must say it was a very interesting discussions and collaboration that I got with several of my colleagues from forensic toxicology concerning validation of high-resolution mass spectrometric methods. Now, we published our ideas in Current Pharmaceutical Design in 2022. So for those interested, I would really suggest to give it a read. But in short, certainly if you're looking into screening methods using high-resolution mass spectrometry, we would suggest a flow looking into a mix of compounds, then looking if your method is selective, sensitive, if there are issues of carryover, matrix effect, stability, and then evaluate via kind of cross-validation of incurred samples. Now, selectivity, we've already a little bit pointed it out, that you can have false positive hits. So you should be aware that when doing the validation, you should have a look into not only sample preparation and analysis, but to include your data analysis as well. If you have blanks, would you have false positive hits that you are missing or you would consider as a positive? That's something to look into. Concerning sensitivity, it's of interest here in screening methods to look at the limit of identification. Verify your identification criteria if they work well. So again, you can do this for a mix, but of course in the screening you have lots and lots of compounds. So maybe each time a new compound arrives, you will have to have a look into the possible limit of identification. Concerning carryover, this is done in most methods, so you have to be aware that the next sample that you will analyze after a really high concentrated sample doesn't have spikes into it, so that you don't consider it as positive. For matrix effect, this is also a classical validation parameter. We should look into the peak area of each analyte in a standard solution versus the same standards injected with a blank sample. Stability is often process sample stability, or of course the screening method. But of course, when you do a validation of a screening method and you can have a mix, you can't do this for all compounds. And that's why certainly in the beginning when you use this method, it's of interest to analyze authentic samples and to have an idea of what your machine can actually do. Now in this slide you can see some examples of experiments that we did using the mix in our validation. And I'm just going to highlight the limit of identification and the matrix effects, because certainly the limit of identification is maybe a term that's not used everywhere. Some people tend to use LOD or LOQ, but for screening purposes, limit of identification can be interesting, as this gives you the lowest concentration that you can monitor by using your identification criteria. So here we've injected several mixes with varying concentrations from 0.5 nanograms per ml to 20. And here you can see actually what the lowest limit of identification is. Now, for some compounds, this may be not low enough. So this is, of course, something that we will then need to look into for further development of the method. For the matrix effect, we just wanted to see if this was concentration dependent or not. So we can also have a look into future, looking into batches and everything, if matrix effect is a problem or not for several compounds. Now, because when you use mixes, you can't do all of the monitoring with the experiments, it's of interest to analyze authentic samples with your screening method. In that way, you can compare the results that you have obtained with your high-resolution mass spectrometric screening method with established methods. In our case, these are LC-MS methods. So we can observe that some compounds, for example fentanyl, which is detected in 0.6 nanogram per ml, can be picked up in our screening method. However, what we also observed when analyzing authentic samples was that some, for example, you see on the right-hand column the MDMA, was not detected because we had such high concentrations. So there was a kind of saturation, and according to our identification criteria, this was not a peak that was of interest. So what we do now is that we are going to inject the same extract, but once 1 microliter and 3 microliter. The 1 microliter, kind of dilutes the sample, which makes it the saturation less a problem. But on the other hand, we have the 3 microliter injection that gives us enough sensitivity for other possible compounds in the sample. So besides method validation, quality assurance is a very important aspect in a routine laboratory. We have to ensure that the system is suitable for doing the analysis, and of course we need a follow-up of the system itself. In addition, we will of course look into the batch and how we can validate the batch, and what about the quality of the samples, but I will deal with that later on. So when we do a system suitability test, we will do this before each batch. We will look into the detector voltage criteria, mass calibration. We will of course look into the log spray and the background level. If there was something wrong and we need to do an action on the machine before it's ready for to be running, we will inject a kind of mix and evaluate this mix via accuracy, mass accuracy of course, retention time, the peak width, and the baseline level, just to ensure that our machine works in optimal conditions. Now we also follow up concerning cleanliness of the MS, its stability and sensitivity and resolution. Now on the right-hand side, you can see kind of sheets that we have developed in the laboratory to have a quality system and to note if we observe some issues in the machine or if the sensitivity, for example, goes down or not. So when we are going to evaluate our batch, we do this via QC injections, and we have a mix containing bromazepam, diazepam, fluoxetine, oxazepam, and trazodone. We will look if in that batch we could identify those compounds in that specific QC run. For each sample, we will spike it with an internal standard, and we will follow up the area of this internal standard. This really is to make sure that nothing went wrong during the protein precipitation or the injection. Now, in general, for the whole system, what we tend to do is we always subscribe for each of our methods in the lab for proficiency testing. And there is one out for new psychoactive substances screening, and we apply this on our HRMS screening method. And I would really recommend this because in this way, you can, of course, it's just a mix that you get and you don't know what's in it, so you have to identify the compounds. And in that way, you can also observe if you're been missing some compounds. So, for example, we missed the dichloroketamine in our system at that time when we did this proficiency test. So then we're going to look, how do we have to include it in our lab library, yes or no? Now this is an example of a real case for which we, again, applied 100 microliter of blood into the system. And as you can see here, we can get a lot of information out of one screening. Now for the GHB, of course, this is not done via the TOF, this is done by another method. We compared the data from our high-resolution mass spectrometric screening with our LC-MS methods that are used to confirm our initial results. What you can see here is that, for example, for the ethylpentylone, we detected that according to our screening method. However, when we wanted to confirm it, we didn't detect it. Now, luckily, of course, there's a lot of literature and there's a Center for Forensic Science Research and Education, because then they came out with, I would say, an alert in which they described dimethylpentylone, which also metabolized to pentylone. So we got the idea, maybe we got the wrong isomer, and we bought the dimethylpentylone. And yes, indeed, we could confirm the compound. So this is really important. You can have a screening that's very interesting, very rapid, but you always have to be aware of the possibility of isomers. And so, again, it's all about setting identification criteria, but sometimes it just won't work. And in this case, it's interesting to be aware of the literature and to find out if there are other compounds out there. So, of course, the proof of the pudding is in the eating, as we say. So once we have this method, it's of course important that it runs with a lot of samples and it's easy to use. So we tested it out for detection of NPSs at different Belgian festivals. Now, we have festivals, for example, with about 400,000 visitors per weekend. And so blood samples that are drawn from people that go to the emergency or to get help at the festivals themselves, they will be sent to our laboratory for analysis. And our drug unit is also often present, and you can see all kinds of stuff. You can see, candy containing cannabinoids or MDMA. You can see pills. We also see a lot of medication, and all types of compounds are observed there. So here you see the results. The bar chart demonstrates the results of the blood analysis, while the table gives us an idea of the compounds that were found on site, so as seizures. What you can observe is that we found a whole range of compounds in the blood samples, and we can actually, via our screening methods, give this information very fast. So I would say just in a short time window, we can already inform the medical doctors which compounds are in the sample so they can act on it. However, if you find an unknown compound, this can of course give you much more work, as detecting those or elucidating the right structure can be a really tough task. So what you can do first is, via a mirror plot, you can look into candidate peaks for possible elucidation. So actually, this approach is a kind of top-down approach in which you select the most abundant peaks from a visual inspection of the total ion current or the base peak intensity chromatogram. So the peaks actually, the most abundant peaks that are not identified via your initial screening system can be found here. Then, via the elucidating unknown software, chemical formulas will be proposed, and this is actually proposed via looking into the accurate mass, kind of double bond equivalence, or also, of course, the spectra, and they will be compared to available databases such as, for example, the drug bank. Once you have this, I would say, elemental formula fit and the structure proposed, then of course you have a tentative identified compound, and this will need to be confirmed via a reference material. To evaluate the way you do your structural elucidation, proficiency tests are also available. This is one from the NF3 Drug Working Group from 2022. Now they will send out 100 milligrams of a white powder, and then of course it's your task to define which compound it is. Now what we observed by using our system is that we found an isomer, but not the exact substance. So again, maybe you just have to be aware of the pros and cons of your system, and if identifying the right isomer is really of importance, then you should think of other techniques to include in your system to be absolutely sure of the identity of your compounds. So in conclusion, the screening method discussed today is really an easy and simple method with an acceptable turnaround time in the lab. The automated data interpretation gives the analyst the possibility to quickly decide in a case which compound should be confirmed. Of course, there's still a lot of work to do, and we have some future perspectives. We want to have a look into the size of the data files, because we have to handle the data for more than 30 years, is very important for us. We also want to do more validation, add compounds to our list, but also analyze more authentic samples to get a good view of reality. Other QA measures can be placed. We're just thinking about a negative mode. We should add an internal standard and have a mix to check before we analyze the batch. And of course, we never know, maybe in the future we will change our method to a semi-quantitative approach. Now, thanks for your attention, and I'm looking forward to the questions, and I'll hand it over now to Dylan. Thank you.
We would like to thank you, Dr. Wiley, for that wonderful presentation, and invite everyone to continue to stay on with us during a brief moment of silence while we transition on to our live Q&A section. Welcome to our live Q&A, and thank you all for staying on. We'd like to thank Dr. Wiley once again for that great presentation and invite you to ask any questions, and Sarah will answer those. So please submit them at your convenience, and I will have her answer them. It looks like we already have some questions coming in. I will go ahead and start with the first question. What was the reason for the missed findings and the samples which contained high levels of drugs, poor mass error, or retention time deviation?
Well, if I remember correct, I think both aspects of our identification criteria were missed. So it can be that you have such a high concentration that the retention time deviation is too big, and so the initial peak will not be picked up by your identification criteria and put in the automatic report. So that's something, again, in the future we'll be looking at just to see if we can adapt this on our automatic report production. However, what we do at the moment is that we inject 2 volumes, 1 microliter and then another one 3 microliter. So we do a kind of dilution of the extract to make sure this is not a problem in our routine work.
Great, thank you so much. We have another question that came in asking a couple of questions here. It says the first question is about the stability that you mentioned in your methods, that it was reducing by 15%. Is that acceptable? And then the second question is, how did you get around it to solve it? And actually, it looks like there's a third. And what was the reason for this reduction in stability?
I think it's about the criteria that we set ourselves in the sense that when we have a sample, so an extract, and we inject it, we accept up to 15% of loss during a certain period. For example, let us say you do your analysis on a Friday. You have to be sure that when you come back to the lab on Monday and there would have been a problem, that you can re-inject your extract without having a huge loss in stability, so that actually your results of your screening would still be adequate. So what we do in the validation is we look into this, I would say process sample stability, and we accept 15% degradation. I think this is something that you observe in lots of international guidelines, but of course it's up to the lab to see if, according to their procedure, this loss is acceptable, yes or no. So that's a part of our validation strategy.
Fantastic, thank you. And we do have more questions that came in. One is asking, in festivals, do you think that LC-MS is a slow and more expensive method when compared to automated immunoassays, which is cheaper and faster?
Of course, LC-MS is slower and is more expensive, and I think it's all about what you want to do in your festival. For us, it worked out fine because we wanted to have the extra specificity of the LC-MS rather than the immunoassays, which can often have, the possibility of a false positive. So we wanted to avoid that. But it's true, the workload it's harder and it's slower. So it depends, I believe, on what the aim is, what you want to do with your analysis, and if, being very fast is more important in your setting.
Our next question is, how often do you see fentanyl and its analogs in your samples?
We do see fentanyl from time to time, but at the moment I don't have any case in which we have found fentanyl analogs. I know they're around, and there's certainly a problem, for example, in the States, but up to date we didn't have a sample that was positive for a fentanyl analog.
Great, thank you very much. The next question is, when looking for carbon-13 isomers of compounds such as amphetamines in samples with very high levels useful, can this alter the dynamic range, and is the approach used in HRMS?
I know that some people will use the 13, the carbon isomers. That's certainly an interesting approach. However, we did not use that, so we went for, I would say, the 3 and 1 microliter samples, just also to observe the very low concentrations of other compounds. So I think, again, it's a choice. It's certainly an option, and I believe this approach is certainly used in higher resolution models.
The next question is a little bit related to that one. And why do you choose to inject 2 volumes, 1 microliter and 2 microliters? Is this not complicated?
Yeah, so that's really a choice we made in the lab. We don't have that large batches, so what we do is we directly dilute a sample, so just to ensure that it can be done in one batch and we don't miss high concentrated compounds.
We just had another question come in, and this question is, in the clinical laboratories, we monitor measurements of uncertainty. Is that an important matrix for you? Furthermore, how do you set analytical performance goals?
So the measurement of uncertainty is certainly something we look into. At the moment, I know it's a qualitative method, and there's certainly also discussion is on that to monitor the measurements uncertainty. I know in the paper, one of my co-authors, Brigitte Desharnais, talks a lot about measurement uncertainty of qualitative methods. So I won't go into this in detail, but just have a look in that paper. There are several options on how to tackle measurements of uncertainty for qualitative methods. How do we set analytical performance goals? I don't know exactly to what it refers. Do you mean more the quality assurance, or the way, that we would look into the machine before we start up an analysis? So we do have certain performance criteria for our machine before we tackle a batch. And then, of course, concerning quality assurance, to look into if the batch is okay, we will do some quality samples. And we set goals into also, of course, does the retention time match? Does the mass, is it okay? Do we see the fragments? And if the quality sample is okay, then we can start off the batch.
Thank you again, and we will go on to our next question. When do you plan on having the retention times in the library for the synthetic cannabinoids?
Yeah, that's something that's really a priority now. So we would like to do that as fast as possible to get those retention times. And certainly, actually, what we want to do is update our library with the high resolution, the high-res NPS library. So in the sense that if we have enough entries, we can send them back to the Danish lab that makes this library, and they will also help us to calculate retention times and to update our method. And then, of course, once we do that, we need to do a kind of revalidation of that method.
That will be great. And the next question is, where do you purchase your standards from?
I believe most of the standards are bought via LGP. We buy most of them.
Thank you, and please keep the questions coming in. We do have Dr. Wiley on for about 6 more minutes. The next question is, how would you evaluate matrix effects in each sample during your routine work?
Well, that's something that we've been thinking about, and I think so each sample that comes in, certainly post-mortem samples, you can have a matrix effect, of course, and standardization would maybe be a good idea to use and to have a vision of the matrix effect, or at least the impact that you can have on your sample.
Thank you very much. And do you still believe that confirmation with a standard is necessary with HRMS?
Well, yes, I do think so. So I really like the aspect of screening, and of course I know you can also quantitate with the machine, but I think the first thing that we observed when we were developing the screening approach is that you do observe false positives. So even with, I would say, the high resolution So I always prefer to have a standard that confirms my result to be absolutely sure when I send in my report for forensic interpretation.
Great, and we have one more question, unless there's any more people that want to send any questions in. And that last question is, do they run two methods, the original method and the longer method, for synthetic cannabinoids, or do they just run the longer method now?
So actually, we still run the shorter method at the moment in our routine work because of the fact that the retention times are not yet updated in the longer one. So once that are set, then we would go for the longer run in our routine setting.
Thank you very much. Is there anyone else that would like to ask another question? We have a few more minutes left. We can wait a couple more seconds, maybe 30 more seconds, to see if anybody else might have any questions, or if you have any comments, please feel free to submit any comments you might have as well.
I would certainly like to thank the audience also for the interesting questions. Always nice, and just to get ideas and to get the method even approved further in the future. So thanks for that.
We appreciate everyone that attended our Q&A session. Thank you very much. And with that, we hope that you all have a fantastic day and appreciate your attendance. Have a good day.